Okay, thank you guys. Welcome back for our last session of the day. We're going to be talking about applying learnings to new frontiers. So with that, I'm going to welcome our uh, moderators or speakers. Great.
All right. Okay, thanks everyone for joining us. Last push of the day before some of us go and rogue out with Peter Frampton. So this is the, I think, one of the ultimate connect the dots sessions. And we're going to start with a really compelling presentation from somebody that really needs not to be introduced. Jill Morrison, you discovered a little something one day, and you're going to share some of that and then how to connect the dots for the future. So please, take it away.
So he's referring to when I was a postdoc at NINDS, actually, where I am now as a program director in extramural. I discovered the gene for Niemann-Pick type C. So this was back in 1997 that the paper was published, and this was before the finishing of the Human Genome Project. So identifying a disease gene was much more complicated than it is now. So that's what he's referring to in my contribution. But now I'm a program director at NINDS, and I'm in the Division of Neuroscience, and I'm in charge of a slew of rare and ultra rare diseases, including the lysosomal storage diseases, mitochondrial disorders, leukodystrophies, and born errors of metabolism. And so I work with the Division of Clinical Research and the Division of Translational Research to move research forward to develop therapies for these disorders. So rare disease is actually what's called an RCDC term. So NIH has a series of terms. Some are very specific to a disease. Others are areas of research where they keep track of the amount of dollars that are spent per fiscal year. So NIH as a whole is very committed to rare disease research. And in FY 2023, we funded $6.9 billion in rare disease research. And I work at NINDS, which our mission is to seek the fundamental knowledge of the brain and the nervous system and to use that knowledge to reduce the burden of neurological disease for all people. And we are also very committed to rare disease research. The majority of neurological disorders are rare, and we have funded over $500 million in research in FY 23. So we have the typical series of funding opportunities that you always hear about at NIH with the R01s, the R21s, but we also have a suite of translational funding opportunities that go with early preclinical development all the way to receiving an IND, and then we have a suite of opportunities for clinical trials. And I'm just going to focus on two areas that are specific for rare diseases. So we developed a program announcement to establish clinical trial readiness for rare and neurological disorders. So we realized that we had, if I can use a pointer, we had the infrastructure for clinical research with what we call NeuroNext, which I will be talking about. We had our suites of therapy development programs, but what we didn't have was an opportunity to validate outcome measures and biomarkers for rare neurological and neuromuscular disorders. So this funding opportunity, which has been active for a few years now, is to actually validate biomarkers and outcome measures across multiple clinical sites. And the goal is being able to show that you have a biomarker outcome measure that will show efficacy in a clinical trial. So as I mentioned, we have a whole suite of funding opportunities in our preclinical, early clinical, and clinical trials. I'm only going to focus on what we call urgent, but I want to emphasize, so this is urgent, We have what's called Blueprint Biologics, which is very complementary to how the urgent program works. That can be for more common as well as rare disease. But the urgent program is specific for ultra rare gene-based therapeutics. And the way we came about developing this was a realization that a lot of advocacy groups were developing therapies through funding proof of concept studies with money that they had raised with academics that they have started collaborating with. However, there was a lack of experience in what is involved in going from proof of concept to getting to a clinical trial. And so the expertise in toxicology, expertise in manufacturing, as well as the need for money. So we developed this urgent program, which is for gene replacement therapies, gene editing, ASOs, so any gene-based therapy, where you have the option to use contractors that we have established contracts with to do the toxicology, to do the manufacturing. And what you as the investigator and the advocacy group provide is the expertise in the disease. So for example, if you're doing AAV, once the vector is manufactured, then your group would do the demonstrate whatever model you have available that the clinical grade virus is working as effectively as you would expect. So what we do is we establish these multidisciplinary teams. We have 5 projects that are active right now. I'm involved in 2 of them, and we meet monthly. And these teams go into great detail on how the project is moving forward. And then we work with contractors that have expertise in the various areas needed for gene therapy. And so the expertise in PK, PD, toxicology, manufacturing, and the investigator does have a choice if they have themselves already a collaboration with a manufacturer. They can use that manufacturer or they can use our contract. So there's flexibility within the program. And then we have the ability to provide you with the knowledge of 25 consultants that we have established contracts with. And they can include things like medical writing, working with you to write your IND, regulatory affairs, and bioassays, et cetera. So the next step was we realized we had this program with the goal that we would quickly move these products into the clinic. But now what do we do once we get this work into where we have the IND? And so we already had an established network for phase 2 clinical trials called NeuroNext. And Dr. Sophie Cho is in the audience. She is my colleague who runs NeuroNext, so she's going to know if I've made a mistake. But hopefully she won't point it out. But this program has been around since 2011. We have a clinical coordinating center, a data coordinating center, and then we have 20 clinical sites throughout the US. So what we did new in 2024 was we decided we wanted to use the infrastructure of NeuroNext to establish a network to do gene therapy trials. So the NeuroNext Clinical Coordinating Center was tasked to establish a gene therapy consortium that would work with in collaboration with the infrastructure of NeuroNext. And so we have established the gene therapy consortium, and here's the name of the colleagues that are well known in the neurological disorders for the gene therapy for neurological disorders. So the idea is that you apply to come into urgent. You work on your preclinical development. You don't have to apply for a grant again. You can go directly into the clinical trial through what's called other transaction authority. And that I would leave to Sophie to explain to you. But it's an internal review process involving NeuroNext that would avoid you having to go through a regular NIH study section again. And the goal is that then you can go straight into a clinical trial. And if your site is a partner next, your site can be added as part of the network. So we try to provide flexibility in making sure that the areas of excellence for a particular rare disease are included. So, and then the last thing I just want to mention is that NINDS is one of the biggest co-funders of the RDCRN, the Rare Disease Clinical Research Network. If you're aware of this, we just received our applications for the next cycle, and they're going to be reviewed in the next couple of months. But this is also an area that we've been highly active in to move rare disease research forward. So thank you.
Thanks, Jill. That was a fantastic presentation that connects the dots with how to fund the important research that we need to continue advancing science forward. So now on to the panel. And I'd like to start the panel conversation with Yulia. You have been, you witnessed firsthand the transition from a vision and an idea of a public-private partnership into the actual establishment of a public-private partnership. So can you help us with some of the perspectives of how to adjust that and how to make that happen, and some of the learnings that we accrued together as we walked through that journey?
Sure. As Deputy Director for the Division of Rare Diseases and Medical Genetics, we're very much, very acutely aware of the difficulties of, doing the road, going the road in the product development alone in a rare genetic metabolic disorders. It is so difficult. The complications are, of course, the first and foremost is that the number of patients available for the clinical development is so small, right? In addition, the natural history of the disease is very poorly characterized for many of genetic metabolic disorders. There are, significant heterogeneity, both, the genotypic and phenotypic heterogeneity. There are different difficulties in understanding and the spectrum of the various rates of disease progression, and therefore it is so critical for the multiple players and critical stakeholders in the product development for rare metabolic disorders to come together. And we're talking about the patients, caregivers, patient groups, the industry, the academicians, and regulators to come together to really have a vision of where we can take the program development from basically, very, very, very basic understanding to the next level, where we can have products being developed in consort with understanding of the natural history of the disease and the biomarkers in those diseases to bring the products that are safe and effective to fruition. So, what is important is to understand the science and to follow the science, and that is understanding what is important to patients, and also it is understanding what's going on in the labs, in the academic environment, and also what are prospective products that are in development that we can take to the next stage of the development in the clinical trial space. So what I can see, critical path program, for example, for CPLD, this is critical path, and the consortia for the lysosomal diseases had done so far is to bring those, critical stakeholders together to understand and chart, what are the important gaps in these diseases and what we can do to advance the product development. That again includes multiple stakeholders. We have patient groups, we have industry sponsors, and we have academicians and as well as regulators on board. It's important to strategize what we can develop first, and sort of build on the knowledge that is being accumulated over time. Thinking about the clinical outcome assessments, thinking about the potential biomarkers, whether it's proximal biomarkers that are metabolites, they're directly being affected by the lack of particular enzyme, but also downstream biomarkers. There are common pathways that we can learn about, from other disease areas as well, in addition to just lysosomal storage diseases, and see how those common pathways might be affected in the lysosomal diseases as well. Try to inform, what are the other potential biomarkers might be, that might be helpful in the product development, and linking those, the clinical progression of the disease, the symptomatology, the diagnostic, potential prognostic biomarkers to the actual clinical disease progression, and see whether or not a particular patient population could be selected for the clinical trials, inform the clinical trial design. What is the patient population? What kind of clinical outcomes could be assessed? What are the clinical outcomes that are meaningful to patient, but also are appropriate to be evaluated in clinical trials as well? Because, obviously the clinical trials are very limited in scope. Which are the clinical outcomes that are, sensitive to change? And then potentially with all of this, creating a very fertile environment for the most efficient clinical trials that we can conduct to inform, and bring the products that are, in most efficient manner to the market.
Fantastic, because that shows you that this is not just about what already exists and the lessons that we can accrue from what has been done in Alzheimer's disease and Parkinson's and Duchenne and Friedreich's ataxia and PKD and tuberculosis, but about how this can be put to good use for the future and for areas of unmet need that have not been addressed yet, and show that it is possible. But we are going to need the data. So, Sophia, can you help us with your perspective and experience in setting up some pretty valuable data resources in the mitochondrial disease space, and how this links to what we've discussed about RDC-ADAP and data sharing, et cetera?
Sure. Thank you. Just a quick introduction. So I'm with Cure-Mito Foundation, and our focus is advancing research of Leigh syndrome. Leigh syndrome is the most common type of mitochondrial disease, and my daughter died from Leigh syndrome as a newborn. It's a fatal disease that usually leads to death in childhood. I work in the industry myself, in statistical programming. So after my daughter died, I really want to bring my knowledge of data into mitochondrial disease community and help from the data perspective. And so with the Cure-Mito Foundation, I'm leading the global patient registry for Leigh syndrome. Our goal, so our registry is around for 3 years. And when we started our registry, we started it with the sort of idea that it's up to us. If we want to see something come out of our registry, if we want to see results, if we want to see something meaningful, and if we want to see this registry really help the community, it's kind of in our hands. We can't collect data and wait for someone to do something, because we've seen so many patient groups, unfortunately, start registries, collect data, and in a few years, there's nothing. And so that's what our thinking was, we don't know if the industry will get our data or be interested. We don't know if researchers will check our data. We have to rely on ourselves. And so that's kind of was our commitment from the beginning. Our registry is done with a Quartz platform, which is using data elements recommended by the NIH, which actually turned out to be really helpful. We created a survey specifically for Leigh syndrome, and there's another general survey for rare disease that Quartz has. And we kind of we've been true to our commitment from the beginning. So now, after 3 years, we have over 350 patients in our registry. It's truly global. We have more than 40 countries in the registry, and it's really all over the world. We have patients from Africa, from South America, from Eastern Europe, from Asia. It's really, really, truly global registry. I think from the US, it's about 35%. So it's really a huge part of it are patients, international patients from everywhere. We published 2 papers. We have posters that we shared at many conferences. We made our data interoperable with CDISC and OMOP, which is CDISC is a data standard required by the FDA, and OMOP is more commonly used by academic researchers, so we have it both ways. We share data with CPATH. In January last year, we started a mitochondrial and inherited metabolic disease task force with CPATH, with the idea that we wanted to bring the broader community. In mitochondrial disease, we have many, many patient organizations. Mitochondrial diseases are complicated. There are many of them. And so there are many registries. Usually they're kind of uncoordinated with each other. So everyone's doing their own thing. And so our hope was that with this MITA task force, we can bring the community together. I think the one thing that's really important about CPATH is that it's neutral. So it creates more trust from the different task holders when you have a neutral partner leading it. And CPATH works with the FDA. The RDCA DAP is funded by the FDA, so that's very important to us. And they have the expertise in data sharing and proper handling of the data and integrating data of different types. So all of that is incredible. And so we're super grateful for CPATH for their support and their willingness to do the task force with us. Our task force now has 21 members. Out of them, 15 are patient organizations, which is unbelievable that we have so many patient groups in the task force. We don't have nearly enough industry, which is a challenge. So if you're here and if you're from the industry, please get involved. We are working on hoping to leverage what data collections we have and come up with a set of common data elements that maybe can be used for the future mitochondrial disease registries and data collections. And we're trying to engage the community to share more data. And it's very inspiring to see what's done in Duchenne, Alzheimer's disease, ALS. So we are hoping to get to the same thing where we can have successful clinical trials. We recently have a few that were unfortunately failed. So our hope is to figure out the endpoints and get to the point where we can have successful clinical trials in mitochondrial disease. And CPATH is amazing and incredible partner in this. So
thank you, and thanks for the very kind words. But the credit goes to you, because this is yet another example of connecting the dots, where there was passion, there was expertise, and there was a vision to make things happen. It is possible to set up data interoperability, data standardization, data sharing when there are open-minded individuals willing to make it happen. And the other thing that is so critical about that example is that it flips the narrative, because we have a great track record of listening to regulators and what the regulators perceive as unmet needs, and then we deploy and build solutions. We have a great track record in partnering with our pharma members and listening to what they have as unmet needs, and then we deploy the public-private partnership model to build solutions. But this is one of the most compelling examples of working with the communities affected by the conditions to make sure that we add value and make it attractive for investment, because by having the data interoperable, having the data shared, having the data analyzed in a way that can start answering those questions about heterogeneity, variability, disease progression, disease characterization, et cetera, that has an effect on the last panel, how to handle uncertainty. And that's how you get pharma partners interested in areas that have a potential to reduce uncertainty, which maximizes the probability of, as is the technical term, technical and regulatory success. So that's a beautiful example. But then I want to go to Christina, because you represent a company that has been a tremendous partner, but you also have met the challenge of caring for therapeutic areas that are somewhat neglected. And you've interacted, and this is a teaser for tomorrow. You've interacted with us in really challenging areas in neonates, and now you're part of this discussion about lysosomal diseases and mitochondrial diseases. So can you share your perspective and your experience in that journey that you have taken with us?
Absolutely. So my name is Christina Costantino, and I'm the Senior Director at CGIC, which is a benefit corporation that focuses on AIR, RARE, and CARE, which is our neonatal group. I am the lead of our programs in lysosomal storage disorders, and we had the proud approval of 2 of our therapies last year, Velminase alpha for alpha melanocytosis and pegaglucosidase alpha for Fabry disease. And I think what we're finding is that even when a solution can be envisioned or is known, especially in a single gene defect, that is not enough to get a solution to patients. We plan against our patient journey because we know that adolescents in our studies will one day be adults making the choice for the product on the market. And so we cannot assume that the environment that we create around a patient in our clinical trials is going to be the environment that they're in a day-to-day basis with this therapy for the rest of their lives. We're also in an environment, as you mentioned before, that we're significantly resource constrained. As we know, investment into this area can be quite limited. Therapies, we would like our therapies to be available globally, which means we need to be thoughtful about our pricing, and we need to be thoughtful about the delivery mechanisms of those therapies. Do we have appropriate care settings? We're working in disease areas that are relatively unknown, not only to regulators, but also sometimes to the patients themselves. And so identifying even what to measure in a natural history study can be quite difficult. And it creates additional pressure because then you want to put everything you can into your phase 3 study because it might be your only shot at collecting enough patients in order to derive an outcome. So I think this is really important to take a holistic look at that patient journey, understand where in your clinical programs you can introduce aspects like home infusion that would enable patients to have ready access later on, understand how to work with regulators, how to work with, and I think this is where the public-private partnership really can play a key role because when you're educating as a single stakeholder to a regulatory body, there's a little bit of skepticism. This is your story, you have a little bit of data, maybe you can get a couple other voices with you in the room. But when you join forces with others, and I think we saw a great example with the Reagan-Udall Foundation in the MPS space, when multiple stakeholders are coming to the table, suggesting a similar solution, there the barrier of education is to some extent lifted. And the promise is that an outcome can then be developed hopefully in enabling accelerated approvals so we can also get drugs to these patients faster, which helps us to save lives in many situations.
Fantastic, and fantastic teaser for the opening session of tomorrow with Reagan-Udall on stage. This shows exactly the power of collaboration and the power of the vision of the Critical Path Initiative. That's exactly what the Critical Path Initiative set out to do. Set up public-private partnerships so that all the stakeholders can work on optimizing the process so that regulators can optimally regulate and companies can optimally develop. Krista, you've joined us and you've lived firsthand the journey from a lot of enthusiasm into a lot of focus in proposing solution candidates and context of use statements, and how that is not a stagnant process, but how it provides the compass to move things forward. So can you share your perspective as you live that journey firsthand?
Absolutely. So I go to Yulia's initial statement about following the science. And so the journey of this life that I chose to embark on was following the science, but the science was going in 70 different directions. And where I thought it would be, boiled down to just a few pathways. Obviously, there's multiple pathways, and again, there's proximal biomarkers since it was the initial focus, and then there's more distal or long-term organ or systemic applications. And so then piling that on top of what Christina mentioned in terms of the patient disease staging and how the patient experience and all of that comes in. So the science, got very, very difficult to not only follow, but also to understand where it was going, where it was coming from in multiple directions. And so one of the things that we had to do in CPLD is kind of take a step back and just utilize the resources that are in place. And that started again with what Sophia mentioned, and that's the RDCA DAP. And in order to be able to identify those pathways and where to make decision making in terms of context of use, we initially have to get the data in an operable place and be able to see different applications of that. And so what we're working on now as we start to embark on multiple disease candidate biomarkers, trying to find those common data elements, it's imperative that the collaborative effort is spearheaded by the data sharing. And the data sharing then allows us to follow the science that is more of a collaborative. We have a lot of enthusiasm about CPLD and about understanding the multiple diseases and accelerating drug development tools. But how we can do that in an actionable way is dependent on getting the data in a format. And so the RDCA DAP has been the foundation of that. And as we move forward, even with lysosomal diseases, but also as Sophia and I have talked about, lysosomal diseases ultimately become mitochondrial diseases. So the rising tide will lift all ships. So the more collaborative efforts in terms of continuing to gain the momentum and the enthusiasm about some of these drug development tools in lysosomal diseases and how that can apply to other diseases. Again, heparin sulfate for MPS was a huge demonstration of collaboration. And we want to do that with multiple biomarkers, with multiple contexts of use across the lysosomal diseases. So that's the journey that we're envisioning. Now, you guys got to help me get there because now we can actually see that it's going to be pretty on the other side.
Fantastic, and fantastic call to action. Now, Jill, there's still in some camps this perception that FDA and NIH are separate worlds that never touch and never link together. However, some of the efforts that you described and some of the efforts that you lead are actually connecting the dots way better between the discovery and the basic science and the clinical science with what happens on the regulatory and medical product development side of things. Can you elaborate on that journey that you also took?
So we as an institute actually have a memorandum of understanding with both CDER and CBER. And so we actually have quarterly calls with the FDA. It provides us an opportunity to bring to them questions that we have about an ongoing project. So, if we're having difficulties in a particular area, they can, give us general advice. And then they also bring to us information on what they're doing and what new insights that they have found, within the last quarter of work. Also, I'm involved with the Bespoke Gene Therapy Consortium. And, the FDA is heavily involved in that effort, and NIH is as well. So in that space also, we work collaboratively. So, I think the two agencies work very well together. And we know we have different roles, but our ultimate goal is the same.
Yeah, which is make sure that we do right by the people that need the developments. Okay, I want to go back to Julia, because you offered a great perspective of the vision to get started, but now that we're moving, can you offer your perspective on how this turned into a forum to assess the real impact, but also the gaps of potential solution candidates, drug development tool candidates like multiple biomarkers, but also in the notion of how we can learn across lysosomal diseases, not just within each individual condition.
Right. Well, you know, rare diseases, rare metabolic diseases come in all different flavors, right? But they're, you know, common concepts, you know, that are true, you know, across, you know, different diseases within the genetic metabolic group of diseases, but also across different therapeutic areas as well. So we envision that we're going to be learning, you know, the science on the other side as well, you know, within the diseases, within the different, you know, spectrum of the diseases of the same kind of pathogenesis, like, you know, mucopolysaccharidoses of different kinds, but also, you know, there are certain neurobehavioral concepts that are being studied in other neurologic diseases that might be very much applicable to the lysosomal diseases as well, particularly those with the neurological outcomes. And that involves not only the clinical outcome assessment tools, but also biomarkers. And, you know, as Krista had said, we do look at the proximal biomarkers. We'll look at the, you know, specific metabolites that are the results of the deficient enzymes, right? But there's, you know, the process, the pathology, the, you know, pathobiology that is happening, it results in the common pathway, right? And there are distal biomarkers that are common across different diseases, whether it's, you know, involvement of different target organs, right? We're talking about the neurons, first of all, but also other systemic organs that might be, you know, very well represented by a qualified biomarker that is sort of the end stage of that pathobiology process. So looking across different therapeutic areas and see what works in other diseases might very much, those learnings will be applicable to the lysosomal diseases as well. So looking at the proximal biomarkers, intermediate biomarkers, but also at the distal biomarkers as well as a common pathway will be very helpful in us to understand that biology process, pathobiology process, and link the clinical outcome or clinical presentation of the disease, the progression of the disease with the changes in those biomarkers. So I'm very excited to hear and be here at the panel, but also during the meeting today and yesterday, and then tomorrow as well, to see the progress that is happening across different areas of medicine, and that the critical path is assuring that this knowledge is transmissible across different areas of medicine as well, that impacts the you know, the regulatory background in the division of rare disease medical genetics, but also have, you know, tremendous impact on the patients with the product availability, or first of all, you know, the clinical trial networks, but also the product availability as a final goal of this exercise.
No, absolutely. And that really connects well the dots with the session that Tarina moderated yesterday. Because it's all about the notion that biomarkers can have really impactful utility beyond just pure diagnostics and fully validated surrogates. There's a full spectrum in between of very tangible, very impactful applications of biomarkers, but we need to, as Krista was saying, define the context of use and understand what the gaps are and what the needs are. But in the spirit of further connecting the dots regarding multiple diseases, so Christina, as a drug developer, we see a trend, and I like that trend of focusing progressively more and more on mechanisms of disease and druggable targets for mechanisms of disease rather than syndromic definition of diseases. Can you comment on that journey and how that's important, especially for rare diseases?
Sure. And, you know, what I'll say is that I wouldn't say it's more important, but I think we need to look at it in two ways. And looking at it two ways enables us to address a broader spectrum of patients, not only those who may be experiencing, you know, their first symptoms or preclinical symptoms, but also patients that have that downstream pathology that maybe is it triggered by accumulation of substrates, et cetera. So most elegantly, we would be able to remediate the gene deficiency. And that's why we're very excited about mRNA editing therapies and therapies like this that I think can correct the deficit, especially if the deficit is known and it is a clear single variant that is of pathologic clarity. That would be the goal. And treating patients in utero would be beautiful, right? And we're starting to see a little bit of this with the study in UCSF with ERT. If we cannot correct the deficit, then the next best would be to replace the enzyme. And I think that's where we are currently. And if we replace the enzyme, how might we get it to the right cell? How might we get it, into the CNS? How do we ensure that that replacement is actually, to some extent, curing the deficit that the patient is experiencing on a cellular level? But that will not be sufficient for a patient that has accumulated stress, has triggered immunological responses, has developed fibrosis. And I think that's where we start to see these syndromes. And very excitingly, in the IAM space, we are starting to see some treatments, especially treatments that you can repurpose to address these metabolic pathways. So we saw some very exciting evidence last week at the Society for the Inborn Errors of Metabolism looking at sildenafil in Leigh disease. So I think this is a good example of how we shouldn't forget that there is some benefit to remediating that at a syndrome level, how those patients are repairing or reducing the progression. But ultimately, our goal is to intervene as early as possible so that the later steps are not needed.
Yeah, okay. You get another word for dot connecting, because you teased up perfectly one of the sessions tomorrow about treating very early as before you're born, and the opportunities that come with that. I'm not going to call them challenges, but pure opportunities that lie with that. But also one thing that you mentioned about not forgetting that symptomatic relief is just as important as meaningfully modifying the disease through multiple mechanisms. And I think, Sophia, that's one thing that I presume you're also thinking about the future in capturing even more longitudinal journeys of the individuals that are part of the MITO data exercise. Can you comment on that?
Yeah, are you asking if I mean, can you repeat the question?
Sure. So as we look at all the potential interventions, right? And as you continue to accrue data on the individuals that are participating, that aspect of the experience with multiple interventions is going to be critical in the longer term. And that goes beyond what Yulia was saying, just the setup of a clinical trial that has a finite duration and how that can add value.
Yeah, absolutely. I mean, we as a Leigh syndrome community, we are looking at different types of treatments that can potentially treat or cure the disease, like gene therapy, drug repurposing. But the treatments that can improve the symptoms, I mean, those are very, very important, because the quality of life of these kids their lives are very, very challenging. So anything that can improve that and make sure that they live not just longer, but also with a better quality of life. I mean, these are very meaningful treatments for our kids.
And that's a really good point for dot connecting, because as your data are interoperable and have CDISC and OMUP structure, that's a great opportunity to capture and link things to the patient-focused drug development conversations that the FDA is always open to have, and include those metrics moving forward, so that it's not just about opening your life, which we greatly appreciate, and that's a show of bravery, but the opening of your life captures data that can really be maximized in utility. Because, I mean, at the end of the day, the topic of today is maximizing the value of every precious data point.
Yeah, exactly. And we had 2 listening sessions with the FDA specifically about our patient registry. And we had that so that we could get feedback from the FDA to make sure we're on the right track. and hear what questions FDA is asking. And I think that was very useful. On our first listening session, many questions were regarding the data quality. How do we assure good quality of the data? And so that helped us. We are doing that. So we are following the FDA guidance. We are doing statistical analysis of the data every month, so on a regular basis, to make sure that we catch data issues, we address them. We looking at the genetic mutation that patients are entering and bringing it to a standard uniform format, because most of the questions we get from the industry are what genes are, what genetic mutations are there. And so, but hearing from the FDA that this is what they are looking for, and they want to know what we are doing to improve the data quality, that was very helpful for us to know that we're on the right track, and as we are going forward.
And the lessons that can be shared with other communities.
Absolutely, absolutely. I actually used to have a working group with Fuse. I recently closed it just because I had too many things. But with that group, we created a few information sheets specifically for patient organizations about what to keep in mind when starting a registry, some myths and facts. And one of the information sheets was taking the FDA guidance on the patient registry and kind of changing into sort of lay language and making concise, and specifically making it for patient organizations. And we shared that very widely. So I'm trying to do that, trying to help other patient groups as much as possible.
Yeah, fantastic. Now, Krista, if you could share the journey that is always important to think about the more specific and more focused aspect regarding a biomarker candidate like NFL and some of the opportunities, but also the gaps that have been identified, and how that's an important component of the portfolio of the CPLD consortium?
Yeah, so it was interesting that we, in the pre-consortium, were at the same time is when the NFL was, neurofilament limit light chain was supported for ALS SOD1. So there was a lot of momentum and a lot of interest there. The problem or the gaps there is that we're starting to accumulate a lot of data in age-related NFL1. With the lysosomal diseases, particularly the pediatric disease, lysosomal diseases, there's a dearth of data for these kids, and there's a lot of confounding factors because of the age-related changes. It's not clear. Now, that being said, we can still learn a lot from other disease states, not only the age-related diseases, FTD and ALS, and even some of Parkinson's and Alzheimer's, but also multiple sclerosis. They're starting to accumulate a large database and age-related, and they've even started to utilize an algorithm for pediatric NFL levels. And so something like NFL is really a ideal candidate to start with. However, one of the additional gaps with lysosomal diseases is that we know from age-related disease that when you have elevated NFL levels, you have a lot of neuronal damage, and in the developing brain, that imposes an additional. We want, going back to Christina, we want to intervene a lot earlier. And when these levels are high, there's more damage that has taken place. That being said, there is some data that has accumulated also in lysosomal diseases that demonstrate a better response to treatment with a higher NFL levels. So that allows us to start to understand what those common pathways are, and potentially what different contexts of use could be for different lysosomal diseases, for different targets. And so it starts this journey that is even more fun, because now we can start to see things and be able to try to circumvent the gaps to have these actionable solutions by learning from other diseases that may seem on the surface as not related, but really learning from that how to go back to the beginning of the pathway and identify intermediate or proximal biomarker that tracks with something like NFL, so that we can utilize maybe a suite of biomarkers for actionable solutions.
Oh, that was a fantastic point, because that illustrates how we fight the tendency of falling into the trap of the last NDA carried forward approach or the last advisory committee carried forward approach. Just because something works in a very specific, if the same biomarker works in a very specific context of use, that doesn't mean that it works in any other context of use for any other condition, but that doesn't detract the value of the biomarker or the value in taking the journey in assessing the different context of use, getting the data in and learning and maximizing the learning from those. Okay, we have about 9 minutes left, and I'm really getting anxious about going to the Peter Frampton concert. So let's close the panel by asking each one of you to give a 1-minute articulation of where you see the public-private partnership efforts that you're involved in the next 3 to 5 years, and what victories you see in the horizon. Let's start with your involvement in MIDO and the task force with RDC ADAPA.
Sure. Our hope, so our task force is big and we have many patient groups. Our hope that we will have industry join our task force, definitely. That we'll have a lot more data shared to the task force, because to do all these incredible things like clinical trial simulation tools and other, we need data. We can't just do it without data. So we hope we'll get a lot more data shared. We hope we can get all of our community united around this task force, and it will grow into something bigger like a consortium, and that we'll have something where we can use this data to really find endpoints, test out different endpoints, design better clinical trials, and we'll have successful clinical trials. So that's what we want to see.
Fantastic. Christina?
Yes, well, I think first and foremost, it creates some momentum and enthusiasm around a common problem that we're looking to solve. And it enables us to work together instead of having multiple parties look to access the same small pool of resources that we have. I hope it also creates better standards and norms where we can reduce variability in any way that will enable us to move a lot faster and with lower risk. And then finally, my hope is that this enables us to generate more therapies. We've seen quite a few examples this year of companies that have had to stop what they're doing or aren't able to move forward, who have fundamental questions or challenges with regulators that I think could be supported by a broader community, such that we give these programs a better chance of success. And that's my dream. I think we can get there.
Yes, agreed.
I certainly echo Christina's statement that there's a lot of momentum here, a lot of excitement surrounding the data sharing and all critical stakeholders being at the table in order to create the environment where the clinical trial that are most efficient are on the table, that we're utilizing data and judiciously using the data to identify those biomarkers that are diagnostic and prognostic, to create those the clinical trials that are most efficient to bring the safe and effective products to the community that is currently have significant unmet medical need.
Fantastic. I'm going to jump to Jill and ask you how you see the partnerships between the NIH and FDA growing in the next 3 to 5 years.
So, a thought that keeps going through my head is that, when you do a gene therapy trial, one of the FDA requirements is the continual follow-up of patients. And I can think of 2 examples where phenotypes have shown up that weren't previously identified in a patient population. So Pompe disease is an example where you treat with enzyme replacement therapy, and then the CNS phenotype becomes more prominent because the patients are surviving better. Or in ex vivo hematopoietic stem cell transplantations with lysosomal diseases where the CNS is being treated, but peripheral neuropathy is being an issue. So I think as we work together, the continual communication and the continued follow-up, I don't think we can forget that once patients have a therapy, even an approved therapy, we have to continue to monitor them to make sure we're fully treating the disease and continuously making progress on even better therapies. So that's I just has been going through my mind while we've been sitting here, and I think that's a goal that we should also set forward.
Fantastic. And Krista, as my CPAP colleague, you get the last word.
The last word. I think that for me as a, physiologist and biochemist, my hope is that we identify multiple candidate biomarkers for multiple lysosomal storage diseases and can develop multiple contexts of use for that. But the important thing is not just the identification or development of those biomarkers, but linking into clinically meaningful endpoints and be able to triangulate the data from biomarkers and context of use with clinical outcomes assessments or other outcomes, so that collective will meet the level of evidence for enhancing opportunities for drug development tools.
Fantastic. Well put. Okay, so with that, we close day 2. Thank you so much. I had a lot of fun. Looking forward to day 3. We're going to keep connecting dots. Thank you so much. And those that are going to join us with Peter Frampton, let's rock on.